Alrighty, as people continue to trickle in, I'm just going to give a soft intro here. So welcome everyone. Thank you so much for joining us today with our August 1st webinar here. My name is Tyler Casey, Senior Marketing Specialist here at Belsera. And we're super excited that so many of you are here today to learn about how the additional context provided by spatial transcriptomics adds deeper insights into disease pathology, our understanding of normal tissue function, and how these can inform the next steps in your research. As a reminder for you, we do have 3 key learning takeaways from this webinar to help set the stage before introducing our incredible speaker. Those are revolutionary techniques, enhanced insights, and expert guidance. Those were all featured on the initial registration page. Please also remember to put your questions in the chat throughout the presentation, as we will have time at the end of the talk for our speaker to answer some of those questions. I do now have the pleasure of introducing Tatiana Kovacevic, who is a Scientific Engagement Manager at Belsera and has a doctorate degree from Cambridge University. Tatiana, take it away.
Hi, everybody. Thank you so much for joining us for this first webinar in our series, where I'll be talking about single cell and spatial transcriptomics and how we can be used in biomedical research, and eventually how we at Belsera can also help you use these novel technologies. So just a brief introduction into the sort of the agenda of today's talk. So first, I'll give a very short introduction into why we're interested in understanding transcriptomics on a single cell level. And then we can dive deeper into single cell sequencing itself, how it works, how does the data analysis work. And then we can even have a little sneak peek into our platform and how you can perform some of the analysis that I will be talking about today on the platform itself. And then in the second part of the talk, I'm going to focus a little bit more on spatial transcriptomics in specifics, again, talking a little bit more about the technologies that have been developed in the recent years, and then also the data analysis, and also, again, a few of the tools that we already have on the platform to do this pretty cool technique. And then finally, I will say a few conclusions, and then we will also, obviously, as Tyler said, have a few minutes for the Q&A. So as we all know, humans are very highly organized systems composed of approximately 4 times 10 to the power of 13 cells of various types, forming really harmonious microenvironments to keep proper organ function and normal cellular homeostasis. And almost all cells in the human body have the same set of genetic materials, but their transcriptome information in each cell reflects the unique activity of only really a subset of genes. So profiling gene expression activity in cells is considered as pretty much one of the most authentic approaches how to probe cell identity, state, function, and response. Bulk RNA sequencing, although it was extremely valuable, is a method of choice for transcriptomic analysis of pooled cell populations that we can get from tissue sections or biopsies, for example. And it really measures the average expression level of individual genes across hundreds or thousands or millions of input cells. And it's really useful to get a global idea of gene expression differences between samples. However, the information about the individual cells is really lost in this bulk approach. So single-cell RNA sequencing has emerged as a powerful tool for investigating cellular biology at really an unprecedented resolution, enabling characterization of cellular heterogeneity, identification of rare but significant cell types, and exploration of cell-cell communication and interactions. So how does single-cell sequencing work? The basic idea includes single-cell isolation and capture, then cell lysis, reverse transcription into cDNA, cDNA amplification, library preparation, which is then followed by next-generation sequencing, and then data analysis. Single-cell capture, reverse transcription, and cDNA amplification are probably the most challenging parts among the library preparation steps. There are many different approaches to performing these steps, and different technologies have been developed that take advantage of many of these different approaches to selecting individual cells. For example, fluorescence activated cell sorting is used by SmartSeq or SmartSeq 2. Fluidigm C1 platform uses microfluidics, and then 10x Genomics' Chromium platform and DropSeq use kind of a subset of the microfluidics approach called microbeads. But in the end, even though there is many different ways how to accomplish it, the key outcome of this process is single cell capture. And particularly in high throughput is that each single cell is captured in an isolated reaction mixture, where ultimately all transcripts from one single cell will be uniquely barcoded with sequences that identify the cell that they come from, and even the individual molecules after all this conversion and library preparation is done. And then after this, the sort of pooled samples can go into next generation sequencing, and we can get the data out of the machines and proceed to data analysis. So analysis of single cell RNA sequencing is another key factor, and pretty much currently the major need to broaden the application of this technology in life and clinical sciences. To ensure the availability of single-cell transcriptome analysis tools, numerous developers have created tools and made considerable efforts. And by 2022, there was approximately nearly 1,000 different bioinformatics tools that tackle some part of this analysis process. And some of these tools we also have available on our 7Bridges platform, and I'm going to talk about it in just a few minutes. But the increasing number of these tools for single-cell transcriptome analysis has really illustrated the importance of analytical methods in the field, but also it means that there's more confusion in choosing the tools for single-cell data analysis. In a simple, very simplified overview, analysis of single-cell sequencing datasets can be divided into several general steps. We have the pre-processing, then something that is kind of referred to as general analysis, and then exploratory analysis. So during the pre-processing steps, the FASTQ files that we have gathered from the machine where the analysis or the sequencing has been performed, they now have to be mapped to the reference genome, and all the barcodes need to be kind of read and sorted into the gene cell matrix. And this is done by different tools. For example, different providers of these platforms provide their own tools. So for example, Cell Ranger is a tool that 10x provides to perform this analysis. So that was for the pre-processing steps. Just a second, sorry. Secondly, we have the general analysis that represents steps that are kind of necessary to perform on the data in order to draw any downstream conclusions from it. First, some additional cleanup is needed to be done. During the preparation of your single-cell suspension, variable cells may experience cell death, membrane damage, or maybe adhesion of several cells due to experimental or technical barriers. And these calls really need to be removed from the analysis. And that can be done by looking at different indicators, such as too high or too low number of genes and transcripts that are expressed. Too high meaning that there may be multiple cells that are captured and kind of read out as a single cell, or too few, meaning that you are maybe looking at an empty call, or a call that only contains maybe some ambiental RNA. So also percent of mitochondrial RNA is another measurement that is used to determine cells that have died and if they contain too much mitochondrial RNA. However, it's important to note that the thresholds for these parameters can really not be standardized for all of your cells, because your cells have very different metabolisms, let's call them. And for example, some cells may be very highly active and they just produce more mitochondrial RNA compared to some more dormant cells. So you can really approach them and provide the same thresholds for these parameters, the same for those kinds of cells. So this is just something that needs to be taken into account. Also, the original expression matrices that are generated via Cell Ranger also cannot be directly used for downstream analysis because the expression levels between cells are really not comparable due to different depths of sequencing, different library sizes. So normalization and scaling of the data is also a very important step in the analysis. Then also single cell sequencing data is highly dimensional with tens of thousands of cells in a really simple and in a sample and thousands of genes that are expressed in each cell. So most genes in each cell belong to housekeeping genes or the genes that are not very much differently expressed between different cells. So it doesn't seem useful to use them to differentiate between your cells. So it's really suggested to use highly variable genes. So the top maybe 2,000 or 3,000 highly variable genes so that you can, on one hand, kind of highlight the biological function of your cells because these are the cells that drive their these are the genes that drive their different functions, but also really greatly speed up the compute speed, which is also something that needs to be thought about. Also, different single-cell RNA sequencing data may arise from different times, different sequencing platforms, and there are really inevitable technological or non-biologically significant effects or batch effects that can arise between these data sets. So many tools have been developed to understand and remove batch effect, for example, Scanorama or Harmony or even Seurat's internal batch correcting algorithms. In addition to feature selection or those highly variable genes, dimensionality reduction is also one of the main strategies for processing really these highly dimensional data. And for single-cell RNA sequencing, 2 rounds of dimensional reduction are generally required, with PCA or principal component analysis dimensional reduction performed first, and then either TISNI or UMAP dimensional reduction performed for visualization. And and then finally, clustering has to be performed, and the complexity of single-cell RNA sequencing data promotes the development of a really wide variety of clustering methods. And there have been papers that have kind of looked into several different clustering methods and found some that perform better than others. For example, Seurat is pretty good in this sort of clustering algorithm, and it's also a very kind of fast tool. So it's, of course, there's some trade-offs, but these are also some kind of considerations that need to be taken into account when thinking about analysis of our data. And then finally, we can do cell type annotation. So we've done all this process of clustering the cells, and now finally we get to assign biological meaning to the clusters that are recognized in the data set. And this can be done either manually by just really looking at the markers that are expressed by your different cells in the different clusters, and then comparing them to known markers of different cell populations, or this can be done computationally, and there are many tools that can do this computationally. Some of them kind of compare your data to databases that are present. Some use some almost machine learning methods to do this analysis. So there's many out there that you can like kind of look through and find what is best suited for your specific needs. Finally, we can also talk a little bit more about exploratory analysis. So once we have done all this preparatory work to look at our data, we can finally ask some interesting biological questions. And first thing that comes to mind when we talk about, in general, RNA sequencing would be the differences in expression patterns between our different samples, or in this case, maybe between our different cells. And differential gene expression can also be done with very specifically designed tools to do it for single-cell sequencing. Also, when there is differential gene expression, there always also comes the gene set enrichment analysis that kind of provides a more larger context to just this list of genes that are differentially expressed. And then also some different, very specific to single-cell sequencing analysis can be performed, like cell trajectory where you can really follow the differentiation path of single cells, where if you're interested in, for example, in developmental processes, that can be a very interesting tool. Cell-cell interactions, you can look at cell cycle analysis and understand the distribution of the different phases of the cell cycle in your sample, if this is of interest to you. And of course, we can also talk about spatial level transcriptomics, but we will talk about this a little bit later in the presentation. But every part of this single cell analysis that I just described, we can really perform easily on our 7Bridges platform. So from aligning the reads using, for example, Cell Ranger, as I mentioned, for the 10x data, or maybe Star Solo or Salmon Alvin for any of the other platforms, to performing maybe some downstream general analysis. Like here, for example, our team have created a workflow that basically wraps all of the steps that I just described for the general analysis. So the quality control, the normalization, scaling, dimensional reduction, and clustering and biomarker identification into one very simple workflow that can very simply be applied to your own data. And I will show you actually in a moment on the platform how we can start, how this analysis looks and how you can kind of use it yourself. And then also we can look into the exploratory analysis on the platform and do, for example, gene set enrichment analysis with tools that are available on the platform, or trajectory analysis, and many other ones that are present on the platform itself. So now I'm going to jump kind of into the platform a little bit and show you how all of this analysis can be performed on our platform. So here I have performed some Cell Ranger analysis for a set of 8 samples of single cell sequencing. And here you can see 8 tasks that I actually started for this specific analysis as batch tasks. So I just batched by sample ID, and this helped me to very quickly start 8 analysis where they can in parallel be analyzed and mapped to the reference genome, and I can just kind of do something else. And then finally, when the analysis is done, I can go into the resulting page of the analysis, and here find all of the outputs that were created by this analysis. I just want to show you here that on the left-hand side, you can see all the inputs that were used for this specific run of Cell Ranger, and there was just a few files that I had to select, the FASTQ files and the reference file, and I could easily immediately start my analysis. Just here on the page, you also have information about the parameters that were applied to this analysis, and you can also very easily explore already the results from this analysis. So Cell Ranger will produce some matrices for us that we can use later in our next step of the general analysis, but we can also already very quickly open this summary HTML report from Cell Ranger right in the platform itself. And we can easily, hopefully soon, see the quality, for example, of our sequencing, and already kind of understand if this is a sample that we are willing or happy to kind of continue using in our analysis. Also, I want to highlight that you can, obviously, our platform is cloud-based, meaning that you can take advantage of all the cloud resources that come with a cloud-based platform. So for any of these analysis, you can select using the size of the instances you want to use that are probably in line with the size of your data set that you want to use. So if you have smaller data sets or maybe some smaller computationally less requiring tools, then you can use small instances and in that way save on costs, or you can expand the instance once you need it. But it really allows you the opportunity to scale with your own work and your own data. Now, as I said, we have created some of these matrices through our Cell Ranger analysis, and we can move on to a second kind of analysis, which would be the sort of what I call general analysis, meaning all of those steps of sorting your data in order to use it later for actual biological questions. So here I again could very easily start this analysis by just providing the matrices for that were created previously, and then I click run, and I would get these output files here. Again, output files for this specific analysis, since it's done with Seurat, are just like an RDS object, and also a report HTML file. And again, you can easily open this HTML file and immediately start kind of understanding the results that you got from this analysis. Also, this is just one kind of way of how you can perform single cell sequencing or any kind of analysis really on our platform. The other way is to use our interactive data analysis environment called Data Studio. And our Data Studio analysis basically allows you to open either Jupyter Lab Notebook or or R Studio analysis, and then you can easily kind of either bring your own code to the platform and run it from there, or you can even use some code that our team has already prepared specifically in regards to single cell sequencing data. But here, actually, I've prepared a small kind of notebook that shows you how you can perform this pretty much similar analysis to what we just saw running a task on the platform. But I just wanted to highlight that you can also kind of fit how the platform is used to different users. So for example, if you are a experienced bioinformatician, you'll probably prefer to work by yourself in R or in Python and write your own code and run analysis like that. But for example, if you are maybe a biologist, for example, when I started doing analysis of my own data, I had no experience in bioinformatics. It was a little bit daunting to kind of jump into the coding world. So in those cases, maybe it will be more easy to get people involved with single cell sequencing by just running the analysis in a very straightforward, easy manner on the platform in the form of a task. But again, this all depends on your own personal use and your own personal way of how you really like to operate. Yes, so as I said, tools such as Arranger Toolkit, Seurat workflows, many other like exploratory analysis tools like scCODA, differential gene expression analysis tools, gene set enrichment analysis. These are already tools that are already on our platform and can easily be just taken out of the box and used on your own data. But we also have an extensive library of R and Python code to perform most of these and many more downstream analysis steps that can be utilized if you want to perform either exploratory analysis, more ad hoc, you have specific tools you want to use, or you just like to do it yourself in Python. But again, as I said, it all depends on how you like to work. And our team of really amazing bioinformaticians and experts will be really happy to consult on kind of any aspect of use of the platform and they can also suggest how to use it, or they can even help you write the code, perform the analysis for you. Also, they are very happy to wrap new tools and bring them to the platform that you may really like to use but are not already available out of the box. So they're a really incredible group of people, and I am like really excited to be able to work with them, and they work really in a great manner. Okay, so now that we talked a little bit about the analysis, I also want to talk a little bit about how we can apply single cell sequencing, or how single cell sequencing has been applied to research until today. So in recent years, single cell sequencing has been widely used in the field of tissue and organ development and has greatly promoted developmental biology research. For example, the single cell RNA sequencing has become really a powerful tool to profile, identify, classify, and discover new or rare types and subtypes of different human organs and tissues, giving more profound information about health and disease in development, immunology, or diabetes, or of course, cancer research, for example. And also in clinical settings, single cell sequencing can also has huge potential New opportunities are emerging in target identification owing to improved disease understanding through cell subtyping and highly multiplexed functional genomic screens that are incorporating single-cell RNA sequencing, and they're enhancing target identification and prioritization. Single-cell sequencing is also aiding the selection of relevant preclinical disease models and providing new insights into drug mechanisms of action. In clinical development, single-cell sequencing also informs decision-making via improved biomarker identification for patient stratification and more precise monitoring of drug response and disease progression. The combination of single-cell profiling platforms and really sophisticated computational methods is driving step change improvements in our knowledge of disease biology and in pharmacology. But of course, despite the great advances and ongoing success, single-cell RNA sequencing and really other single-cell omics modalities are not applied in situ, resulting in the loss of information about how cells interact and organize across the tissue landscape, which could really be crucial to understand the underlying biology of these cells and niches. So cellular organization in tissues is really closely linked to biological function. For example, in cancer biology, tumors are known to be highly heterogeneous, and understanding the different niches of the tumor and what their characteristics are in space could really be of huge value in understanding varying responses to therapy. Of course, the basics of observing gene expression in space are really not novel, and they have been used for many years with, for example, RNA fluorescent in situ hybridization technologies. However, these are restricted to only querying a few genes, and you have to know which genes you're looking for in order to look for them. So it is a biased approach to identifying transcriptomes or genes in situ. The modern concept of spatial omics or transcriptomics, whereby combination of new methodologies allows querying of the whole transcriptome in space, was first introduced by Stahl and colleagues in 2016 and has since been commercialized by 10x Genomics as their Visium platform. But this specific method relies on capturing polyadenylated mRNAs that are released from tissue sections onto a barcoded array surface or a slide. And different spatial transcriptomics methodologies cover various aspects regarding the number of detected genes, resolution, and whole transcriptome analysis. And these methods can be broadly classified into several groups depending on kind of what the readout of the target is. And how the spatial context is obtained. For example, sequencing-based methods rely on whole transcriptome sequencing based on spatially barcoded RNA. And this is where spatial transcriptomics or the 10x Genomics Visium kind of falls under. Despite the advances in this next generation sequencing-based methods, most of them lack single-cell resolution or really have relatively low transcript counts, which reduces their sensitivity to low abundance transcripts. Then secondly, we have probe-based methods. They require counting of barcoded probes of known targets, and the nanostrings GeoMx platform falls under this category. Then we have imaging-based methods that depend on repeated imaging cycles to obtain the final readout of known targets. For example, CosMx from Nanostring falls under this, or MERFISH or MERSCOPE, which combines fluorescence in situ hybridization with combinatorial labels to enable sequential rounds of hybridization and imaging. And the serial images are then decoded using error-robust barcodes. And this method does give subcellular resolution compared to sequencing-based methods. However, imaging is like way more computationally heavy compared to the sequencing methodologies. So it's all, of course, about what you are looking for in the end. And also in imaging-based technologies, the field of view of your tissue is relatively limited. And then finally, we have image-guided spatially resolved methods, which select and isolate single cells in regions of interest based on microscopic images, followed by single cell sorting and state-of-the-art single-cell omics sequencing. And the earliest example of this is laser capture microdissection, but there have been more recent developments in this field, like niche-seq or fun-seq, for example. And when it comes to spatially resolved data analysis, similar steps really need to be taken that are discussed previously for like more traditional, let's call it single cell analysis. You have to align the reads to the reference, but now there also needs to be a component of interpretation in the spatial barcode as well. After that, data needs to be normalized, scaled, performed dimensional reduction, and clustered. Secondly, techniques that are based on imaging have to contain many complex pre-processing steps to go from images to this gene spot matrix, such as, for example, filtering to remove any noise in the images, aligning the images from different hybridization and imaging rounds so that the kind of spot that you are reading out is in the same place, so you can assign actual meaning to your codes, and segmentation of cells. But platform manufacturers usually release toolkits to kind of do the pre-processing steps with their respective technology platforms. So for example, 10x has Space Ranger to align your spatially resolved data, or VisGen, who commercialize Mirfish technology. They also have their own like visualizer suite that helps you analyze their own data. On top of that, some more specific data processing steps also need to be performed. And previously, we talked about the resolution of some of the available spatially resolved tools. It's really not single cell. It's more like several cells. And a common issue in spatial transcriptomics is just this. So the detected expression in an aggregation of a heterogeneous set of cells within the spot that you are reading out, which may impact kind of the efficiency and accuracy of identifying your cell subpopulations. So the spatial decomposition process can be done through various deconvolution algorithms, can address this issue, which is to disentangle this mixture of RNAs and subsequently predict the portions of each cell type in one spot. Gene imputation is also another challenge for spatial transcriptomics data analysis, which is the task of to infer lost gene expression information due to dropouts or low count numbers. And this is also a common issue in like imaging-based spatial data analysis. And here you can see just some of the kind of commonly currently used tools for these steps. Now we can also move on to the more exploratory analysis part of the analysis that is made possible with the development of spatial transcriptomics. So a goal of a spatial transcriptomics experiment may be to identify spatial features like anatomical and microanatomical structures. And to identify structures, existing algorithms can group transcriptomically similar spots or cells in an unbiased way to reveal spatial patterns of gene expression. Newer methods have been developed to specifically leverage spatial data and identify features like tissue domains. And these are like base space or this NEST tool that is described here on the slide. Alternatively, rather than identifying heterogeneity among cells and spots across the sample, one might search directly for genes that show biased or non-random spatial expression patterns. And again, numerous methods for detecting genes that vary spatially have emerged over the past few years, with some implemented in popular tools like Surat and Giotto, and some being more specifically developed for this goal, like spatial DE, for example. Another third common goal in analyzing transcriptomic data is to infer cellular interactions based on expression of ligands and receptors, or kind of in a more easily understood terms, the cell-cell interactions in your data set. And this can be done by different tools like cell to location. Again, Giotto has its own internal algorithm that does this, or maybe specific algorithms for this like cell phone DB or MISTI. Really, a range of techniques are required to infer biological processes from spatial transcriptomics, technique-specific methods to prepare the data, to identify a region corresponding to single cells and tissue regions from transcriptomes alone, to infer genes that may not have been profiled by spatial transcriptomics, but instead by another modality, and to identify spatially mediated biology, such as cell-cell interactions. And again, these are just a few of the tools that we already have on our Seven Bridges platform. But I also want to highlight that our platform is data type and tool type agnostic. So as long as your tool of interest that you would like to use in the cloud is either Python or R-based or is a command line tool, it can be brought to the platform and you can take advantage of all of the kind of perks that cloud-based environment brings. And also currently our team is working on a kind of comparative study between three toolkits, or Seurat, Giotto, and Scampi, specifically for spatial transcriptomics data, to see which ones kind of perform the best, and to create kind of a more streamlined pipeline of spatial transcriptomics analysis. So these novel methods have been applied in the field of oncology already, cancer immunology, and developmental biology. And here you can see just a table that lists just some of the papers that are released using spatial transcriptomics techniques from a review that was published last year. For example, diverse heterogeneity of tumor-associated macrophages and their role in influencing tumor microenvironmental landscapes was better understood with spatial omics profiling, and also metastatic liver cancer microenvironment was found to undergo really spatially defined reprogramming, specifically in a subtype of macrophages which harbor enhanced metabolic activity and contribute to cancer metastasis. So these were just a few findings that were made by single-cell transcriptomics approaches. So to conclude, single-cell sequencing has really proven as one of the transforming technologies in life sciences over the past decade. The development of high-throughput single-cell RNA sequencing and the computational tools that make technology accessible and applicable in almost all applications in life sciences. Despite all the great promises, of course, one major kind of drawback of single-cell RNA sequencing is the lack of this histological spatial information, which led to rapid development of spatial transcriptomics and the different technologies that allow you to have this yet another additional layer of information on top of your single-cell data. On one hand, even though it has substantially decreased, the price for single-cell RNA sequencing is still quite high, and it's an expensive technology, not to mention the spatial transcriptomics, which is even more expensive, which really limits its use in a more routine diagnostic tool, and is still primarily used as exploratory, in exploratory phases of clinical research. And another limiting factor to using these technologies are often complex data analysis steps. Making the technology available to users who do not have a background in bioinformatics can also really help propel it to the forefront of clinical diagnostics. And also these analysis toolkits are really computationally demanding, and using cloud-based technologies is probably or cloud-based environments is probably where the field is going in terms of being able to process such large data sets. And I really hope that I could kind of show you how our platform works and how Valsera could really, and the Seven Bridges platform could really help in addressing some of these kind of questions that arise from using these very complex tools. So thank you very much, everybody, for your attention, and I will be very happy to take any questions.
Awesome. Thank you, Tatiana. What a wonderful presentation filled with tons of great information. So thank you for sharing that. As Tatiana said, we will now take some time for questions. So if you have any questions, feel free to throw them in the Q&A. Tatiana, I'm going to read out this first one to you, and then I'll have you answer it, and then we'll continue down the list here. I have heard dispersion is a better metric for identifying significant genes since it is less influenced by normalization techniques. Can you comment on this at all?
Um, I I really don't think I can comment since I mean I'm not really familiar with it. So I think, but if this is like a novel kind of approach to it, it may be that the field has a little bit moved on in how it looks at the sort of highly variable genes and like what is taken into account for clustering. But yeah, I'm not sure. Sorry.
Okay. Hello, thank you for your inspiring presentation. Could you provide some insight useful in the very first stages of project planning? For example, we want to add scRNA-seq to our experiments, and we want to sequence 50 samples of 10K cells each. How can we get an approximation of the amount computing resources we would need for the analysis of such an experiment? Also, is there a way in which we can assess and advance the amount of data we will generate?
So 50 samples of 10,000 cells each. I mean, I'm not sure how to calculate the sort of actual numbers of how to kind of process all this data, but it sounds like a lot, a lot of cells. It's 500,000 cells if my math is correct. So I would say that your best bet would be to use like a cloud-based method. For example, I personally, in my PhD, I used maybe my data set had a little bit less than 100,000 cells, and I was really struggling to run this analysis on our university's cluster. So it may be that it could be best for you to kind of upgrade to a cloud-based environment. I would also say that starting a single cell experiment is kind of, to me at least, it was really daunting. So I think it would be great to like sit down and really think about if single cell sequencing is the kind of like the tool of best use for you. And also once you're there, then also planning the experiments and experimental setup, I think is also crucial in how you approach this kind of your questions because of the different batch effects and how you're going to perform batch correction. It's really important to think about how you're going to collect your samples, how you're going to sequence them, and then later, so that later in the analysis, you have the least amount of kind of wrangling of the data to do. I hope that was helpful.
Is there any best practices for choosing clustering parameters?
Yes, this is always, I think, a question when it comes to single cell sequencing to me. I personally think that you need to iterate a few of the resolution settings and then see what best fits with your data. And you really need to pay attention how the clustering is being done and what is the algorithm finding the difference based on. So you have to kind of push it to the limit where you are finding the most clusters, but not finding clusters that are, for example, due to your technical variability, like the different batches or something like that. So you should probably iterate it a few times, look at different resolutions, and then see what fits best with your data set.
Thank you for the wonderful presentation. From your personal experience, would you trust at all marker genes in spatial transcriptomics due to large presence of gene dropout?
Um, I think to an extent you can trust the marker genes, but I do agree that there probably are different kind of like as I was talking about the different tools that can help you evade the dropout issue or any of the other kind of technological caveats that come with the spatial single cell. But I think to an extent you could trust it. I mean.
My team is working on niche use cases that will probably require some custom scripts and references. Can you help us with this development?
Yes, this is something that our team can definitely help you with. Um, They are currently, as I said, already working on several projects for either spatial or single cell sequencing, where we use really custom tools to help our clients do kind of all the ideas that they have, kind of funnel them into actual analysis. So I think our team is very well equipped to help you, and we will be very happy to maybe discuss it like offline. But yes.
We want to use large public data sets. Can that be something that we can use on the platform?
Yes, so we have a few ways of how you can bring public data sets into the platform. We already have access to like the TCGA through our sister platform for academic use called Cancer Genomics Cloud. But we also have ability to kind of bring in, for example, SRA data into our platform, and then you can continue using the data from there and all the different tools that come with the platform. So yes.
Awesome. And then the last question I'm seeing is, can I use proprietary software developed by the makers of single cell technology instruments on your platform?
Yes, so we already have a few of those tools that are present on the platform, like for example, what I said, Cell Range or Space Ranger, Cell Ranger from 10x. We also have some nanostring toolkits on the platform. The only caveat there is that you have to have the license to use those tools. And if you do have the license, then you can use them on the platform. But also we are always very happy to kind of interact with the company who you are working with, if we do not have their tools on the platform, to see how we can come up with kind of a solution to bring the toolkit on the platform, and then also for you to use it.
Awesome. And then I had one more question come in. Can you comment on whether pseudo bulk or methods developed for single cell are better for DE analysis?
So I personally, when I was doing single cell sequencing, I used a pseudo bulk approach, because to me it looked like the best way to utilize the best of both worlds. So to use single cell to really get down to the very best, like defined cell subpopulations of cells, but then within that I can use pseudo bulk to kind of apply or compare the clusters as they are, rather than individual cells. So for me personally, I think the combination of both is the best approach to kind of what I was looking at, which was looking at different conditions. For example, a condition where I treated some cells with one drug versus another, or things like that. So I think pseudobulk is probably good when you are looking for these like more general kind of changes in your clusters. Yeah.
Awesome. Perfect. Well, I don't see any other questions. So it looks like we covered everything for today. And thank you all for joining today's webinar on single cell and spatial transcriptomic capabilities in biomedical research. And we hope that you will join us for our next upcoming webinar with the topic soon to be announced on October 3rd. As just a reminder, today's key takeaways really included revolutionary techniques, enhanced insights, and expert guidance that Tatiana shared with all of us. To close, Valsera is committed to powering your discoveries and serving as the foundation for collaborative, data-driven research. If you are interested in learning more and diving deeper into and past what Tatiana presented, please feel free to submit a demo request on valsera.com to continue the conversation. We thank you for joining us today, and we'll see you on October 3rd. Have a wonderful rest of your day.
Thank you, everybody.